Welcome to week 9 of October 17th. Last week you took your midterms and a lot of people did pretty exceptionally well. The average was a C, which makes sense for an average grade. But I did have a few people get an A, and I even got one person received 100%, so that was great. Go ahead and check anything you missed with the answer key that I posted under resources. We're going to do just a few selections from chapter 10, 11, and 12. We have a lot of book left to cover, so we'll just be doing 3 to 6 top sections of each chapter from now on and kind of lumping the main concepts together. So chapter 10 is about the origin and diversification of life on Earth. We won't be talking about the origin of life so much as the diversity of life. And so life is defined by the ability to replicate and by the presence of some sort of metabolism. We're going to focus on sections 4 through 5, 7 through 9, 14 through 18. Species are the basic units of biodiversity. We'll talk about evolutionary trees that help us categorize that biodiversity. Not so much macroevolution, but we will talk about diversity of life in an overview sort of way, and then the subsequent chapters will be specifics on plants, animals, fungi, bacteria. So the species is the basic unit of biodiversity. These are several different species of plants. So the question we want to answer is, how do we know when to classify two individuals as members of different groups and when to classify them as two individuals who are members of the same group? So how do we know that it's the same species or not? We can't solely go based on looks. So on the left, we're showing you two different versions of the same thing. We have Beyonce Knowles and Cristiano Ronaldo, both human beings. Homo sapiens is the scientific species name. So we generally distinguish between different kinds of living organisms. In the middle we have a rose versus a daisy. Similarly, we lump other organisms into the same group and recognize them as different versions of the same thing. Red roses versus yellow roses, or chihuahuas and Dalmatians. How do we know when to classify two individuals as members of different groups and when to classify them as two individuals who are members of the same group? So the examples here on the right, the flowers and the monkeys, are kind of showing you that those two things look similar, but they are different species. Whereas on the left, we have Beyonce and Cristiano, and they are the same species. So what is the dividing line? It can't just be looks. Biologists use the word species to label different kinds of organisms. And according to the biological species concept, species are natural populations of organisms that interbreed with each other or could possibly interbreed, and that cannot interbreed with organisms outside their group. So this all depends on reproduction, not on the physical appearance. Notice that the biological species concept completely ignores physical appearance when defining a species and instead emphasizes reproductive isolation, which is the inability of the individuals from two populations to produce fertile offspring with each other, thereby making it impossible for them to exchange genes. So let's clarify two important features of the biological species concept. You should definitely know these. First, it says that members of a species are either actually interbreeding or could possibly interbreed. This emphasis means that just because two individuals are physically separated, they aren't necessarily in different species. So a person living in the United States and a person living in Iceland, for example, may not be able to mate because of the distance between them, but if they were brought to the same location, they could mate if they wanted to. So we do not consider them to be reproductively isolated. Second, our definition refers to a natural population. This distinction is important because in captivity, occasionally individuals may interbreed which would not interbreed in the wild, such as zebras and horses. That's that example shown here. So even though the zebra and the horse made this Zorse offspring shown in this figure, that would never naturally happen. So we would still consider a horse and a zebra two different species. So what are the barriers to reproduction that keep species from interbreeding? We have prezygotic and postzygotic barriers. You should know this. Remember, an egg that has been fertilized by a sperm is called a zygote. So that might help you remember. Pre means before the zygote, and then postzygotic means post after the zygote. So prezygotic would be pre-fertilization to make the zygote. Postzygotic would be post-fertilization to make the zygote. Listed here, prezygotic barriers are individuals physically unable to mate with each other. It's just logistically won't work out. Or if individuals are able to mate, the male's reproductive cell is unable to fertilize the female's reproductive cell. Postzygotic would be mating produce hybrid individuals that do not survive long after fertilization. So they may never fully develop and become born. Or if a hybrid offspring does survive, such as the mule above, they're infertile or have reduced fertility. You may have heard of mules. Mules are infertile. So that would be a postzygotic barrier because the offspring can't go on and keep reproducing to make more and more mules. So that would not be a species. And we just talked about that. Those are prezygotic barriers. Barriers would include, for prezygotic, courtship rituals, physical differences, physical or biochemical factors involving the gametes. The gametes are, again, those eggs and sperm. So if they can't come together, the egg and sperm can't come together, then you have a prezygotic barrier. Postzygotic barriers occur after fertilization, generally prevent the production of fertile offspring from individuals of two different species. And if an offspring is viable, that offspring is called a hybrid. So know that. These barriers are responsible for the production of hybrid individuals that either do not survive long after fertilization, or if they do survive, are infertile or have reduced fertility. Mules, for example, are the hybrid offspring of horses and donkeys. And even though they can fully survive and live complete lives, they cannot breed with each other or produce offspring. So because they're infertile, that would be a postzygotic barrier. So that tells us that donkeys and horses are two separate species. A Zorse is sterile, suggesting what type of reproductive barrier? Think about it. Go back if you need to. The answer is number 3, postzygotic. So obviously the egg and the sperm of the horse and the zebra came together and fertilized and made this Zorse that fully developed and came out. But because this hybrid is sterile, it cannot go on to produce offspring of its own. We know that the horse and the zebra have a postzygotic barrier. That means they're two separate species. So this is reminding you species are defined as those two things that we've gone over. So keeping track of a large group of anything requires an organizational system. Many libraries, for example, catalog their books into different classes and then further subdivide each class. You know, you got fiction, you got romance, all of that, and then you might have divisions based on the author's last name, for example. Or another example is on your computer. You store your electronic files with virtual folders and subfolders that can be organized by name, size, or type. So the strength of the Linnaeus system is that it is hierarchical. That is, each element of the system falls under just a single element in the level just above it. So we go from broad domains to specific species names. In the Linnaeus system, species is the narrowest classification for an organism. The name of every species within a genus is unique, but many different species may be in the same genus. Okay? Similarly, many genera, that's just genus plural, are within a family, and many families are grouped within an order. Orders are grouped within a class. Classes are grouped within a phylum, and as Linnaeus originally set it up, all phyla are classified under one of three kingdoms, which would include the animal, plant, or he called it the mineral, but we don't use three kingdoms anymore. We changed that system to a four-kingdom system, so we have protists, plants, fungi, and animals. So know those four kingdoms that we use today, not necessarily the ones that Linnaeus used. Today, many of the species classifications that Linnaeus described have been revised, and some of his designations, such as the mineral kingdom, for example, have been left out. Also, all of the kingdoms are now classified under an even higher order of classification, the domain. But Linnaeus' basic hierarchical structure remains, and all life on Earth is still named using this system, with all organisms belonging to one of the three domains. So you need to know this order that's broad to narrow, that's domain to kingdom to phylum to class to order to family to genus to species. You should know those three domains. You should know those four kingdoms. You should know how a species is named with that genus and specific epithet. And you should understand that it's italicized and the genus name is capitalized. Bacteria, archaea, eukarya. So here's where we talk about the macroevolution part. So how do new species arise? How do we get all these species running around? Biologists don't really have a clue about how many species there are on Earth. Estimates of the number vary tremendously, from low estimates of 5 million to high estimates of 100 million. Biologists do, however, know the process by which they all arose. The process of speciation, in which one species splits into two distinct species, occurs in two distinct phases and requires more than just evolutionary change in a population. The first phase of speciation is reproductive isolation, which we've already discussed, through which two populations come to have independent evolutionary fates. The second phase of speciation is genetic divergence, in which two populations evolve as separate entities, accumulating physical and behavioral differences over time as they become adapted differently to features of their environment, including predators and types and abundances of food available. So this is why understanding evolution, mutations, adaptation, natural selection from the previous two chapters is pretty important. So you should understand these steps to speciation and what it is. Speciation is genetic divergence, which is number 4. But for genetic divergence to happen, we have to inhibit the gene flow between the two populations, and we have to have natural selection be the mechanism that's acting to adapt and result in evolution of those two different species. Speciation can also occur among populations that overlap geographically. I think this one's harder to wrap your head around than allopatric, but you do need to know allopatric and sympatric are two different types of speciation. Anyway, when speciation occurs in populations that overlap, that type of speciation is called sympatric speciation. In the case of animals, it's rare for populations of the same animals to become reproductively isolated when they coexist in the same area. So this method of speciation is relatively uncommon in animals, but among plants, it is common. And I'm not going to go into the specifics of how that happens in plants. But the take-home message here is speciation is a process by which one species splits into two. It can occur by a combination of reproductive isolation and genetic divergence. Know those two things. Charles Darwin proposed and documented that species could in fact give rise to new species. With Darwin, the classification of species acquired a new goal and a more important function. In The Origin of Species, Darwin wrote, quote, "Our classification will come to be, as far as they can be so made, genealogies," end quote. That is, Darwin proposed that the classification of organisms would resemble family trees, or genealogies, that link parents and offspring over long periods of time. With these words, Darwin was the first to link classification with evolution. Linnaean classification involved placing organisms within groups as a function of their apparent similarity with each other. That would be the domain, kingdom, all of that we just went over. The modern incarnation of Darwin's vision of classification is called systematics. It has the broader goal of reconstructing the phylogeny. Phylogeny is defined as the evolutionary history of organisms. So through systematics, all species, even extinct ones, are named and arranged in a manner that indicates the common ancestor they share and the points at which they diverged. So that's the key to understanding these evolutionary trees that I'm going to show you. That they are all rooted in a common ancestor and they branch off. If we talk about a tree, they branch at points in which they diverge from each other. So how to read an evolutionary tree. A phylogenetic tree not only shows the relationship among organisms, but also presents a hypothesis about the evolutionary history of species. At the beginning of life on Earth, there was the first living organism, one that could replicate itself and that had a metabolism. So that was about 3.4 billion years ago. Then a speciation event occurred. We had one species in the entire Earth, but then a speciation event occurred, so we went from 1 to 2. After this event, the population of the first living organism split into independent evolutionary lineages. So every time you have a large split, all of those ancestors will start forming their own branch versus the other branch. So back then, with the first speciation event, the phylogenetic tree had its first branch, and there was biodiversity. It's important to remember that phylogenies are hypotheses, and like any hypotheses, they are subject to revision and modification. Over hundreds of millions of years since then, speciation events continue to occur. Today, the tree has branches with millions or possibly tens of millions, depending how you draw it, of the tips that represent all species on Earth, and it doesn't stop there. Even today, speciation continues to add new branches all the time. So evolutionary trees reveal the evolutionary history of species and the sequence of speciation events that gave rise to them. So if you look at this picture down to the left, the red arrow is pointing to a common ancestor of all the animals shown. So there's a common ancestor to fish, birds, human, rats, and mouse. And at that point, the common ancestor of all of those, a speciation event occurred and the ancestral species split into two. One that gave rise to fish and the other one that then gave rise to everything else. Well, there's another common ancestor to birds, humans, rats, and mice. There was a split there as well, a different speciation event. So one branch went off into birds and the other branch went into humans, rats, and mouse. So those are all mammals, right? Well, there's another speciation event. So one's going to eventually go off into humans and the other branch is going to go off into rat and mouse. The branches of an evolutionary tree can be spun around the point at which they split and still remain accurate. So no matter how I draw the final ordering on top, as long as those branches are in the right order, then my tree is accurate. The history of life can be visualized as a tree. The tree reveals the evolutionary history of all species, and here's an important part, the sequence of speciation events that gave rise to them. Many human traditions have a sacred tree of life. The Bodhi tree that the Buddha sat beneath for 7 years to achieve the ultimate truth is the tree of enlightenment. The apple tree in the Garden of Eden is the tree of knowledge. The history of the relationship between all organisms that constitute life on Earth is another tree of life. The trunk and branches of an evolutionary tree represent ancestor-descendant relationships that link living organisms with all life that has ever existed on Earth. The evolutionary tree of life can be thought of as one giant tree. But as a practical matter, biologists often study only particular branches. These branches can be illustrated as big trees or little trees and can be expanded to include whatever organisms the biologist is studying. The tree might, for example, include all animals or just the rodents. What trees tell us is which groups are most closely related to which other groups. Okay, that's important. Which groups are more closely related to which other groups? One of the most interesting revelations of this tree type of thinking is that despite appearances, certain things are closer together than we would think from appearances. So fungi, such as mushrooms, yeast, and mold, are more closely related to animals than to plants. And we know this by looking at their DNA. So I've talked about this before. We can compare how close two organisms are by their DNA. So we share 99% the same DNA with a chimpanzee versus an eel. So if we put that on an evolutionary tree, we would have branches closer together to the chimpanzee than to the eels. And we kind of see that here. So let's see, animals are in the top right. Those are the furthest away from bacteria, but are fairly close to protists. So if I want to say what's closely related, are plants more closely related to archaea or protists? Think about that. The answer should be protists. If I want to say, are fungi more closely related to animals or to plants, it would be animals. So going back to this picture, evolutionary trees tell us many things, but they do not tell us which groups are most, quote, primitive and which are, quote, advanced. This property of phylogenetic trees can serve to undermine some of our most sacred beliefs, such as the one that humans are the pinnacle of evolution. Many trees can be drawn to support this idea, but notice that if you rotate this tree around one of its, we call that a node, where it diverges as a node, you can get a number of different trees. That's just kind of just to let you know. Reading an evolutionary tree reveals which groups are most closely related and approximately how long ago they shared a common ancestor. Know that. But how are evolutionary trees that might hypothesize historical events that happened in the case of the cat-dog split 60 million years ago, how are they constructed in the first place? I kind of gave you a hint of this, but until recently, these trees were assembled by looking carefully at numerous physical features of species and generating tables that compared these features across the species. So this table is a simple example of such a table. With only 3 features, this table shows a clear split between the characteristics of the lion and the hyena on one hand and the wolf and the bear on the other hand. So this is showing you the 3 qualifiers that they used to make their tree. And honestly, you could go out and do this yourself. I mean, you could come up with some sort of a trait and make your own tree. Maybe I'll have you do that for your homework. For most of the 20th century, that means the 1900s, biologists classifying organisms would also use 50 or more characters to generate a tree. But now, starting in the 1980s, biologists began using molecular sequences of the DNA. The rationale for this approach is that organisms inherit DNA from their ancestors, and so as species diverge, their DNA sequences also diverge, becoming increasingly different, which we've talked about, and that's because of the accumulation of those mutations. As more time that passes following the splitting of one species into 2, the difference in their DNA sequences becomes greater, because there's been longer for mutations to build up, right? By comparing how similar the DNA sequences are between 2 groups, it is possible to estimate how long it has been since they shared a common ancestor. So here they're showing you species 1 and 2 are more similar to each other than they are to either species 3 or 4. That's suggesting that they shared a more recent common ancestor. So they're going to be put together with a common ancestor, whereas species 3 and 4 has their separate common ancestor. And then all the species come back to one common ancestor down toward the bottom of that fork. Evolutionary trees constructed by biologists are hypotheses, so they could change. They represent an attempt to describe which groups are more closely related to which other groups. I wanted to talk about this because I don't think a lot of people know this. There have been several mass extinctions on Earth. If the past is a guide to the future, we know this: no species lasts forever. So that's showing you here, you probably know about that mass extinction. So extinction of almost all dinosaur species, and actually 3/4 of all species on Earth. That happened approximately 90 million years ago. But they were equally large and one even bigger extinction event that you probably don't know about. So there's one around 200 million years. There's a mass extinction called the Great Dying at about 230 million years. We have one at 380 and about 450. So you should know that mass extinctions are due to extraordinary and sudden changes in the environment, and that there are background extinctions that occur all the time, mostly as a result of natural selection. So the difference between mass extinctions and background extinctions is that they have different causes. So mass extinctions are due to the environmental effects, such as the asteroid that brought about the dinosaurs' extinction. As a consequence, nothing more than bad luck is responsible for the extinction of species during mass extinction. Fit and unfit individuals alike perish. Whereas with background extinctions, those are going to be unfit species are going to die off, whereas fit species are going to flourish in a given area. Of the 5 mass extinctions during the past 500 million years, okay, so know that there's been 5 mass extinctions in 500 million years. The most recent one is also the best understood. So you should know about this one. 65 million years ago, a massive asteroid smashed into the Caribbean near the Yucatan Peninsula, Mexico. The impact left a crater more than 100 miles wide and probably created an enormous fireball that caused fires worldwide, followed by a cloud of dust and debris that blocked all the sunlight from the Earth and disturbed the climate worldwide for months. So you should know that wiped out almost all the dinosaurs and 3/4 of all species on Earth at that time. Again, that was 65 million years ago. That was one of the 5 mass extinctions in the past 500 million years. Okay, so you should know the 2 types of extinctions and what causes them. So there's 3 large groups that we divide all living organisms into, and then we funnel down into more and more specific groupings based on that Linnaeus classification kingdoms. That's showing you this here. You see how Proteus is labeled a couple of times? They have problems categorizing Proteus because they're kind of weird. A 4th group of diversity and important biological qualities are the viruses. And we don't even technically call viruses life at this point. They're not even included in the tree of life at all because of that. They're not considered to be living organisms. So they can replicate, but they only have metabolism by taking over the processes of another organism. So they can't reproduce their own DNA, and they can't perform their own metabolism at all. Like, they have to live inside of a different cell. So they're not considered life at this point in time. But some scientists view viruses as living and want to put that into the tree of life. So we might see some large revisions happening to this 3-domain system in the future. But for right now, the most commonly accepted tree of life suggests that after the origin of life, the following sequence of events occurred. 1, bacteria arose from the first self-replicating metabolizing cells. Okay, so here's our ancestral bacterium. First single-celled organism, first life on Earth, 3.4 billion years ago. And then there was a split between the bacteria and a line that gave rise to the archaea and eukarya. Alright, so here's bacteria. Here's going up to this next split into archaea and eukarya. Then, step 3, the fusion of a bacterium and an archaea-like prokaryote gave rise to the eukaryotes. So blue over here, this would be the eukaryotes, okay? So you have an archaeon, which goes in the archaea, common ancestor, and then we got a eukaryote, which goes in the eukarya, common ancestor. Remember we talked about endosymbiosis theory with mitochondria and chloroplasts? This is kind of that sort of an event we were talking about. That common ancestor between bacteria and archaea, that split happened, remember, 3 billion years ago. And then this common ancestor between archaea and eukarya, that domain split happened 2.5 billion years ago. Sorry, gonna go back. Alright, the archaea exhibit tremendous diversity But what we do is we divide them into 5 groups based on their physiological features. You should know these 5 things. Number 1 is thermophiles. Thermo meaning heat, phile meaning lover. So they are heat lovers. They live in very hot places where no other organisms can survive. Halophiles means lovers of salt. Though they live in very salty places. We have high and low pH tolerant. Normally things like a pH around 7, but these can live in really acidic or really basic environments. We have high pressure tolerant. Those are the ones that are found deep sea. They can be found as far down as 2.5 miles below the ocean surface, where the pressure is almost 6,000 pounds per square inch. So that's compared with an air pressure of less than 15 pounds at sea level. That's pretty crazy. And then number 5 are the methanogens, which are anaerobic, meaning they don't need oxygen at all, and they produce methane gas. So they're adapted to life in extreme environments, and we talked about the 5 categories in which they can survive. The last domain is eukarya. That consists of the 4 kingdoms you're most familiar with, which is plants and animals, but also fungi, which is funguses, and then protists. So all 4 of these kingdoms are made up of eukaryotic cells. They have membrane-enclosed nucleus. They have organelles. And each kingdom is almost entirely multicellular. So protists have some single-celled, but mostly the eukarya are multicellular. Mostly. There are 3 kingdoms of eukarya that can be seen with the naked eye, that is plants, animals, and fungi. All are made up from eukaryotic cells, which means they have a membrane-enclosed nucleus, and each kingdom is almost entirely multicellular. A 4th kingdom, which you may not have heard about, contains protists, which are often too small to be seen by the naked eye, so you need a microscope. And they're kind of like a grab bag that include a wide range of mostly single-celled eukaryotic organisms. So this includes amoebas, which you've probably heard of, paramecia, and then algae. Those are all protists. Okay, so eukarya are mostly multicellular except for the kingdom protist, which is weird. As we noted earlier, biologists now know that the protists are not a monophyletic group. We didn't really talk about that, actually, we just skipped that. But we're increasingly splitting them into multiple kingdoms within the eukarya. So we're learning more and more about the protists as we go on and learning better how to categorize them. So of the 1.5 million named species on Earth, the majority are eukarya, with about half being insects. So remember those estimates that there might be up to 500 million species on Earth, but we've only ever named 1.5 million of them. So what we know the most about are the eukarya. There's a lot we have yet to learn about those other 2 domains. And half of the things we have named are insects. This is more of a result of interest and bias of biologists than a reflection of the relative number of actual species in the world. So I want you to understand that the most diverse is the bacteria, and we have mostly bacteria, we just haven't named so many of them. So moving on to chapter 11, talking about animals now. We're going to talk about animals and their diversity. Sections 1, 3, 13, 18 through 20. So we'll define an animal and the key distinctions that divide the species up. We'll talk about vertebrates. Specifically, we'll talk a lot about mammals since we're mammals. As you just learned, eukarya is the domain that includes the kingdom animals. So it's just one branch of the eukarya domain. So what is an animal? Surprisingly, considering the enormous differences among even the few animals shown above, just 3 characteristics are genetically sufficient to define animals. We know that animals eat other animals, so they acquire energy by consuming other organisms, right? Plants do not do that. They make their own, but we have to consume other organisms. We know that animals move, whereas plants are stationary, right? So all animals have the ability to move at least at some stage of their life. And they're multicellular. So every single animal is multicellular. That means it has many cells. They consist of multiple cells, and they have body parts that are specialized for doing different things. So know those 3 characteristics that define an animal. And then within the category of animals, we have 4 key distinctions. More than 2/3 of the almost 2 million species on Earth that have been identified are animals. With such a large number of animals to organize and identify, it is useful to divide them into a few large categories based on their evolutionary relatedness. Alright, so you need to know this chart. You need to know these 4 things and what splits into which categories. So each of these distinctions hinges on a particular adaptation and the question of whether an animal descends from an ancestor with that adaptation. So while you're looking at this chart, I'm going to go ahead and keep reading the next 4 slides about these 4 key characteristics, so you can go read them later if you want. So characteristic 1, the question is, does the animal have specialized cells that form defined tissues? If an animal has no tissues, a tissue is a group of cells with a common structure and function. Think of our lungs versus our heart. Those are 2 different tissues. If an animal has no tissues, then we do not need to ask any further questions because we know we can put it in the phylum Porifera, better known as sponges. I don't need you to know porifera, I need you to know sponges. So sponges do not have defined tissue. Instead of having tissues, they're just pretty much clumps of similar cells, and they don't really have coordination or activity among those clumps of cells. All other animals form a group with clearly defined tissues. Humans, for example, have highly specialized cells like skin cells, muscle cells, sensory cells. So then we go on to question 2. All animals with defined tissues develop in a shape that has some sort of symmetry. Is that animal developed with radial symmetry or bilateral symmetry? So know what these 2 are. Radial symmetry describes animals with a body structure kind of like a pie. So you could cut it any way and you get symmetry. That would be like jellyfishes, corals, sea anemones. Together they're grouped under cnidarians. So know cnidarians, and know that includes like jellyfishes and corals. Because of that radial symmetry, they don't really have a front end or a back end. Again, think of a pie. There's no front or back. It's just the same all the way around. So it's possible to make multiple slices cutting through the center to divide the organism into identical pieces. In contrast, organisms with bilateral symmetry, such as humans, cows, scorpions, etc., they have a left and right side that are mirror images. So these animals can move adeptly, searching for food and avoiding predators. Okay, so we've gotten sponges and cnidarians out of the way with those first 2 questions. Question 3, during development, does the animal's gut develop from front to back or back to front? So early in the evolution of animals, this is about 630 million years ago, a major split occurred that allowed us to separate bilaterally symmetrical animals with defined tissue into 2 distinct lineages. The protostomes, so know that, protostomes, which can be translated as mouth first, or front to back, mouth to back, and the deuterostomes, which means mouth second. These names refer to the way the gut develops, from front to back or from back to front. In protostomes, the gut develops from front to back, so the first opening that forms becomes the mouth of the adult animal, and the last opening becomes the anus. In deuterostomes, the gut develops from back to front, so the anus is the first opening to form, and the second is the mouth. Alright, so protostomes include a lot of groups. We've got flatworms, annelids, which is worms. Those are roundworms versus flatworms. Mollusks, more roundworms, and then arthropods. Whereas deuterostomes include chordates, which means there's a spinal cord, versus echinoderms. We'll talk about that next. Alright, so among the bilaterally symmetrical animals that are protostomes, there's an important distinction between animals that molt, which means shedding an exoskeleton and replacing it with a larger one at regular intervals during their life, and animals that grow by adding the size of their skeletal elements in a more continuous manner than among the molting animals. Okay, so roundworms and arthropods grow by molting. Within the protostomes, we had to break them down further. We have roundworms and arthropods that grow by molting, and then we got mollusks, flatworms, annelids that grow by adding continually to the body. And that's these 4 questions here, so you should know these. Alright, so if we look down at that bottom at this chart we've already been talking about, this evolutionary tree, the invertebrates is everything but the chordates. Okay? That means they have vertebrae, like our spinal cord has those vertebrae discs in it. So everything else doesn't have those. In spite of the evolutionary rationale of organizing animal diversity, there is a distinction that may be commonly used, which is whether the animal has a backbone or not. So invertebrates, including sponges, all of those animals that are not chordates, they do not have a backbone, whereas vertebrates are animals that do. Invertebrates are the largest and most diverse group of animals. They are diverse, but they also include 8 separate evolutionary lineages with diverse body forms, body sizes, habitats, and behaviors. See that on the far right? Chordates have vertebrae. They're the only ones that are vertebrates. Alright, so all animals probably originated from an ancestral protist. There are 4 key distinctions that you need to know. Again, invertebrates are animals without a background. They are the most diverse group of animals. Within invertebrates, arthropods, they make up 75% of the animal species on Earth, just so you know. So the arthropod phylum is so large that even if we excluded all the insects, it would still contain more species than any other phylum. So here are arthropods. We have insects, but we also have millipedes and centipedes, arachnids, and crustaceans. Those are all arthropods. Echinoderms are the vertebrates' closest relative. Echinoderms are invertebrates, but they are the closest to the vertebrates evolutionarily. This includes sea stars, sea urchins, sand dollars. They are covered by spiny plates, but because they are disterostomes, their gut forms from back to front, which makes them more closely related to humans, which is kind of strange because they look like they should be more closely related to mollusks, but because of their gut development plan, they're more closely related to chordates. So I'm showing you that in the top right. Alright, lastly, the phylum Chordata, which includes vertebrates, which is animals with a backbone. That's what we care about. Okay, so chordates aren't all vertebrates, but all vertebrates are chordates, which might confuse you. Think about it. Probably need to draw a picture. Okay, we have terrestrial vertebrates. They're all tetrapods, which mean they have 4 legs, and they can be amniotes or non-amniotes. It is the presence or absence of the amniotic egg that allows us to distinguish 2 large groups of terrestrial vertebrates. The non-amniotes would be amphibians. The amniotes would include reptiles and mammals. And so that's what we're going to talk about next. Amniotic vertebrates. That includes lizards and snakes, turtles, crocodiles, birds, both living and extinct, and then off on their own we have mammals. Soon after amniotic vertebrates appeared, 2 different evolutionary lineages began to diverge. One of these is the mammals. So here's the common ancestor here between mammals and all the other guys. The group to which humans belong. The other lineages is the one we've been calling reptiles, but that also includes birds. Birds have about 9,700 species. They are merely one branch of the reptile lineage. And they also include snakes and lizards, turtles, crocodiles, and alligators, etc. The characteristics that hold the bird, crocodile, dinosaur group together are mostly similarities in bones and in their DNA. Reptiles are amniotes and thus have amniotic eggs. So you are familiar with the hard-shelled eggs of birds, and some lizard, snakes, and turtle eggs also have hard shells. Other species in these groups have eggs with a parchment-like shell. Most people find it confusing that birds are grouped with lizards and snakes, turtles, and alligators, and that confusion is easy to understand. After all, birds are covered by feathers and they fly, whereas the other reptiles have bare skin and do not fly. There is an important physiological difference as well. Birds are endoderms, which means they use the heat produced by their cellular respiration to raise their body temperature, whereas reptiles are ectoterms. They bask in the sun to raise their body temperature. You should know that. The most apparent difference between reptiles, including most dinosaurs, and birds is the presence of feathers, though. But the fossil record reveals that feathers actually evolved before birds. So we know there are dinosaurs, which everyone knows those are reptiles, but we know that they had feathers. So they're actually, like, possibly dinosaurs are more closely related to birds. Are the mammary glands and the hair. There are mammals that lay eggs. Monotremes are mammals that lay eggs, but they also produce milk. Today there are actually only 5 species of monotremes that survive. One of them is the platypus. The remaining 2 lineages of mammals are the marsupials and the placentals. They're both viviparous, but the newborn young of marsupials and placentals are quite different. Marsupials are called the pouched mammals because the females of most marsupial species have a pouch where the young complete their development following a short period of embryonic life in the uterus. Whereas placental mammals, which include humans, take their name from the placenta, which is the structure that is responsible for the transfer of nutrients, gases, waste products, etc. between the mother and the developing fetus. That has dense capillary beds that send projections into the wall of the uterus, where the fetus is surrounded by blood from the mother's circulation. So even within mammals, we have these 3 different categories: monotremes, marsupials, and placental. Definitely know those. Based on their modes of reproduction, which animal resemble their reptilian ancestors the most? The answer is monotremes, because they actually lay eggs. Alright, to know the defining features of mammals and the 3 different kinds. The evolutionary lineage to which humans belong, the primates, originated about 55 million years ago. So know that, 55 million. Again, primates evolved 55 million years ago. We can get an idea of the appearance of the ancestral primate from the modern species of tree-living mammals called prosimians, which is a group that includes lemurs. Many of the anatomical characteristics of humans and other primates can be traced to our arboreal origin, meaning living in the tree. That includes our forward-directed eyes and our binocular vision that allows us to judge distances accurately. Also, we share shoulder and elbow joints that allow our arms to rotate. We have 10 fingers and 10 toes that allow us to grasp objects. Those are all traits we inherited from our arboreal ancestors. Okay, so within the group of placental mammals, which are chordates, which have those other 4 characteristics we talked about as well, we have the primates, which can be further broken down into prosimians, monkeys, and apes, and humans are included in the ape category. So humans are part of the anthropoid lineage of primates. You don't really need to know that, but it includes the New World and Old World monkeys, both of which have tails, and then the apes, which lack tails. Okay, so that's the difference between apes and monkeys, is the tails. People call apes monkeys and monkeys apes all the time. It kind of drives me crazy. They're 2 different things. Among the apes, gibbons and orangutans live in pairs or alone, while gorillas and chimpanzees live in social groups that consist of 1 or more adult males and several females, the ancestral social structure for human societies. Within the apes, genetic and anatomical characteristics show that chimpanzees are the closest living relative of humans. Human and chimpanzee genes are very similar. Their sequence differs by only 1%. So we only have a 1% difference in. A third of our genes are exactly like base to base to base, meaning those 4 letters are identical. So the ordering of them is identical. The amount of genetic difference between human and chimpanzees indicates that the chimpanzee that we see today and the modern human lineage separated only 5 or 6 million years ago. So know that date. So how do humans differ from chimpanzees? Well, you probably think you differ a lot from a chimpanzee, which makes sense. we've evolved a lot of different characteristics, different adaptations. So one is we are bipedal, meaning we walk on two legs all the time. We are bigger than chimpanzees, and we have a bigger brain. We're brainier than chimpanzees. When we trace the appearance of human characteristics through the fossil record for those 5 to 6 million years, we find that humans did not gradually become bipedal, big, and brainy. The three characteristics evolved one by one. So first, being bipedal evolved. So our oldest human ancestors were bipedal, but they were about the size of chimpanzees and had about the same size brain. The next thing that happened is that the brain volume increased. And finally, an increase in body size was accompanied by a further increase with the size of the brain. So why did the first bipedal humanoids, why did they start walking on two legs all the time instead of on all fours like chimpanzees? What are the advantages of walking on 2 feet? Well, it's actually pretty significant. So shifting from walking on 4 legs to walking on 2 legs requires changes in several parts of the skeleton, so the way our hips are. The evolution of bipedalism was not a simple process because of that, but it was the first change in the human lineage. It appears to set the stage for all the changes that followed. Anyway, the primary advantage of bipedal locomotion or walking for early humans was probably energetic efficiency. Bipedal locomotion at walking speed uses way less energy than walking on all fours, and efficient walking could have been a valuable adaptation if the human lineage was moving out of the forest onto the savanna, while the chimpanzee lineage, as we know, remained in the forest. Another thing that I've read about is how being in the savanna, meaning like grasslands instead of the trees, walking upright allows you to look up over all of that grass and constantly be able to see in front of you, whereas a chimpanzee couldn't really see what was happening if it was down on all fours in that grass all the time. This is the evolutionary history of humans as we know right now, so the fossil records that we have available right now. Of course, there's fossils that I'm sure we haven't found yet. But evolution in general doesn't follow a straight line. I think that's a problem that people have. They think like, oh, well, you say evolution and mutations are kind of random, but then you say there's a straight line. No, I don't actually say there is a straight line. With any speciation, we have a lot of species that aren't fruitful and they kind of die off. Remember, we always have that background extinction rate. So the evolutionary history of humans did not follow a straight line. Since first appearing around 4 million years ago, there have been many branches to the human evolutionary tree that just died off, whereas Homo sapiens won out and we're still around. So about 3.5 or 4 million years ago, several bipedal groups known as Australopithecines appeared. You may have heard of Lucy. She's an Australopithecine. So this is one of the first bipedal humanoid species. But they were no larger than chimpanzees, and they had the same brain volumes. So this is how we know we can date, remember the carbon dating, we can date these skeletons to 3.5-ish million years ago, specifically 3.2 million years ago, and we can say, hey, they were bipedal, but they were the same size, and their brains were the same size, so we know what evolved first was the adaptation of bipedalism. The fossil known as Lucy was the first of many Australopithecines to be discovered. So since then, it was found in the 70s, and it's called Lucy after the Beatles song Lucy in the Sky with Diamonds, actually. But since Lucy was found, we've found other fossils for Australopithecines as well. So Lucy, though, she was only 3 feet tall and weighed no more than 60 pounds. And the Australopithecine group to which Lucy belonged lived in grassy habitats with scattered trees. Their hands and feet retain the curved finger and toe bones that are characteristics of arboreal animals. So when you go to the zoo and you see chimpanzees, like, you know, you look at your thumb right now compared to the other 4 fingers, your thumb's way apart. But on our feet, our toes really are all close together. But you see the chimpanzees climbing trees, their big toe is offset like our thumbs. So Lucy would have had offset thumbs and toes. Following the appearance of bipedal primates, a second branching episode in human evolution originated from the Australopithecines and produced the earliest species in our own genus, known as Homo. So Homo sapiens is our species, but the genus is Homo, meaning man, sapiens meaning wise man. So there are other species that were around that are ancestors of Homo sapiens. These earlier humans had brain volumes about twice those of chimpanzees, but with little increase in body size. These species had markedly smaller teeth than the Australopithecine ancestors, a change that might indicate they had started to use tools instead of their teeth for the initial preparation of food. Stone tools are found in the same deposits as the fossils of Homo habilis. Homo habilis actually means, habilis means like working, I believe, something close to that in the Latin. So working man. So he made tools. And we found tools with those fossils. And that was about 2 million years ago. Homo habilis and the species may have been the first to use tools. Up to this time, all of human evolution had taken place in the southern and eastern parts of Africa, but about 2 million years ago, one branch, represented by Homo erectus, migrated out of Africa and spread through Eastern Europe and Asia. So that's where we finally start seeing fossils of humans in continents besides Africa. And then they migrated out of Africa and spread through Eastern Europe and Asia. But then we see Homo ergaster remained in Africa. So there were Homo, genus Homo, species. Some remained in Africa, some went and traveled further away into Asia and other areas of Europe. And so those branched into 2 totally different species. Additionally, branching episodes in human evolution gave rise to several species of advanced humans, which include Neanderthals. Those are the stereotypical cavemen that you see in movies. Those are the Neanderthals. And a lot of people think that Homo sapiens came from Neanderthals. But that's not true. So Neanderthals are their own branching species that kind of died off. We didn't come directly from them. Remember, this is not a linear event. There's a bunch of branches of species that come off and kind of die. Homo neanderthalensis, I said that wrong, sorry. Homo neanderthalensis and our own species, Homo sapiens, are 2 different branches. So this coincided with an increase in body size to approximately the height and weight of modern humans, and an increase in brain volume to nearly twice that of the earlier ancestors. In addition, the body form of the species that evolved during this episode looked like modern humans with shorter arms and longer legs. You might be wondering, how do we know how big their brains were? Because brains don't fossilize, just the bones do. Well, what they do is they can figure out the volume of the brain in the skull. They can figure out the size there, that big empty hole in the skull. They can fill it up with volume and figure out how big it is. And then that's how they can figure out the brain size. It's pretty straightforward, actually. Hopefully that was informative to you. Humans, forward-looking eyes, hands and feet with 10 fingers and 10 toes. Our shoulder and elbow joints that allow the arms to rotate are characteristics retained from our tree-dwelling arboreal ancestors. So know that the early ancestors of humans left the trees about 4.5 to 5 million years ago, and they became bipeds. That was the first thing that changed. And then know the 2 subsequent changes of the brain volume and the size. About 200,000 years ago, a new human species branched off from Homo ergaster lineage in Africa, and that's the first modern Homo sapiens. So about 200,000 years ago in Africa, our species Homo sapiens arose. 200,000 years is a ballpark figure based on a combination of molecular and fossil evidence. Then about 60,000 years ago, a small group, probably no more than 100, of these modern humans left Africa and ultimately spread across the earth. So that's 60,000 years ago that we see human migration out of Africa. And there was this really cool exhibit from the Smithsonian that came through the Science Center here in St. Louis. And they tracked the genetic changes in the DNA of humans as we spread from Africa throughout the entire globe, so we could trace where exactly in time humans had gone when they left Africa. We could show that they first were in Europe, they spread down into Asia, Australia, across to North America, and then South America. Really fascinating. So one path you can see here turned west after leaving Africa and spread into Europe, but then a second path turned southeast, spread into southern Asia and throughout the Indo-Australian archipelago to Australia. So there used to be land masses that were closer together, so you could kind of have a land bridge is what we call it to get down to Australia. And then the third migration is the same sort of a land bridge thing. The third migration of humans moved northeast, populating northern Asia. They crossed the Bering Straits, that's a land bridge that existed 15,000 years ago, and spread southward through the Americas. So the climate 15,000 years ago was not the same as now, and all of the placements of land on the Earth are not the same as they are now. In fact, the continents have been shifting throughout Earth's entire history. So know that Homo sapiens evolved in Africa about 200,000 years ago, and then left about 60,000 years ago and started migrating. Again, Homo sapiens existed at the same time as other human species, but they died off. There were two other species at the same time Homo sapiens was flourishing. Those two died off about 30,000 to 12,000 years ago. Very interesting stuff. Hopefully for some of your homework I can find some videos about the fossils that we use that we've found of all these humans throughout the last 4 million years. Now we've been talking about the diversity of animals, now we need to talk about the diversity of plants and fungus. We're just going to go over sections 1 and 13, explain what a plant technically is, and the relationship between fungi and plants. Plants go under that domain eukarya. These are 3 things that make plants. Plants create their own food, plants don't really move, sessile means they don't move, and then plants are multicellular. So they obtain their own food, they carry out photosynthesis, which means they need sunlight, so plants are always growing toward the sun. They're stationary. Because they're stationary, how do they have sex? I mean, there are male and female plants, so sex is a challenge for any organism that is anchored in place by its roots. But plants have a sex life that is very different from animals because of that. Humans and other animals have no haploid stage in their life other than gametes, but plants do. We're not really going to talk about that because it's confusing. But the haploid stage is the one that allows the male and female plants to reproduce, even though they will never meet because they cannot move. Another way that they are able to have sex is through pollen and fruit. So those are the egg and sperm cells, are pollen and fruits and stuff like that. They get carried around by insects and other like small birds, and so that they can get to each other even though they can't physically move themselves. Another stationary living challenge is resisting predators. But plants have adapted different ways to defend themselves. One is thorns. They also use chemicals such as, think poison ivy. Plants have their own different categories. I'm not going to go into the specifics of them, but the earliest plants were the first multicellular organisms to live on land. These are simple, non-vascular plants. Vascular meaning tissue. So plants have tissues, just like we have tissues. They just form different kinds of structures. So non-vascular plants had no tissues and they had no vessels in them, sort of like our blood vessels. They had no vessels to transport water and nutrients. So the first question when you're categorizing plants is, does the plant have vessels to transport water and nutrients? The answer is no. Then we're looking at the earliest branch of plants, which is non-vascular plants. The subsequent evolution of land plants was a series of characteristics that made them increasingly independent of water. The evolutionary tree of plants shows these stages clearly. First, the development of vessels to conduct water from the soil through the plant. So that is the first question. Does the plant have vessels to transport water and nutrients? Then seeds were the next evolutionary event. Seeds provide nutrients to get new plants off to a good start. So does the plant produce seeds, yes or no? If it's a no, then we have vascular seedless plants. Ferns are the quintessential example of vascular seedless plants. And finally, flowers. Does the plant produce flowers and fruit, which allows insects and birds to spread the plant's male gametes to get to the female plants? So the third question was, does the plant produce flowers and fruits? If it's no, then we have gymnosperms. If it's yes, we have angiosperms. So angiosperms have flowers and fruit. All the flowering and fruit plants that you see are angiosperms. Plants that have cones and stuff like that, fir trees, those would be gymnosperms. So know those three questions in those four categories. What are the three main challenges a plant must overcome? We just talked about it. The answer is number 4. Because they can't move, they have the issues of getting to the sunlight, sex, and predators. As you walk in the woods, you see a flowering plant. You immediately know that it is which category of plants? The answer is angiosperms. Fungi make up their own kingdom within the eukaryote domain. Most fungi are multicellular, most of them can't move, and they are decomposers. So that is what defines a fungus. They're decomposers that acquire energy by breaking down the tissues of dead organisms. They are sessile, meaning they're anchored in one place. They can't really move around like animals do. And then they have cell walls made of chitin. Fungi have cell walls containing chitin, which is a chemical that is important in producing the exoskeleton of insects. So when we talk about fungus are actually more closely related to animals than plants, that chitin is one reason for that. They kind of have their own sort of exoskeleton. Although they were originally thought to be plants lacking chlorophyll, the fungi turn out to have little in common with plants. And in fact, when we look at their DNA, it reveals that the fungus are more closely related to animals. So all these three things are eukaryotes, but fungi have all the basic cellular components you would expect to find in a eukaryote. They have a nuclei, mitochondria, cytoskeleton, organelles. They also have cell walls, kind of like plants, only plants have cell walls, but the cell walls, instead of being made of cellulose like plants, they're made of chitin, which is more like the exoskeleton of insects. Fungi are most likely arose from a unicellular, flagellated aquatic protist more than 500 million years ago. So we got fungus around 500 million years ago. There have been close to 100,000 species described. The most commonly encountered types are yeasts, which are single-celled fungus. Most fungus are multicellular, but yeasts are single-celled. We use yeast in cooking, obviously. There are mushrooms, that's a category. And we have molds, such as penicillium, which is the source of the antibiotic penicillin. We have black bread mold, all sorts of molds. So fungi are eukaryotes, eukaryote meaning they have a true nucleus with a membrane around it, and they have organelles. They have the chitin, which is their distinctive feature. They're more closely related to animals than plants, and they live as individual cells as yeast, but most of the fungi are